You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you. Hello, this is Richard Jacobs with the Future Tech and Finding Genius podcast. I have Iliyan Iliev, Assistant Professor of Immunology and Medicine at the Weill Cornell Medical College. And we're going to be talking about not the microbiome, but the mycobiome, I guess, the fungal microbiome, which will be super interesting. So, Iliyan, thanks for coming. How you doing?
Yeah, great. Thank you for the invite, Richard.
Yeah, I guess in multicellular creatures like us, we have a microbiome of bacteria. We have a mycobiome of fungi, yeasts, and things like that. I guess we have a parasite microbiome and maybe a virome as well. Is that right?
Yes. So all those are inside of us or on our bodies. What we are interested in is more like the single organisms that are out there, and fungi are special interest of the lab.
Okay, so why this? Why your interest in fungi or fungi? Where did this come from?
So it's very random, as many good things that happen in science. So I'm a mucosal immunologist, so I look at the mucosal surfaces. And many years ago, we have been looking before even those deep sequencing methodologies started. So there wasn't a microbiome or anything like that. So we will use basically different primers to look for specific bacteria that we know they're in the gut because we isolated them. And we'll look specifically in different inflammatory conditions and diseases that target the gastrointestinal tract, such as Crohn's disease and ulcerative colitis. So in this set of primers, by PCR, we detected basically a fungal message. There was only one pan-fungal primer, which people would use actually to look for contaminations in tissue cultures. So we applied that into that context, and we found this fungal message constantly coming. So I kind of filed that in my head, and then have been doing my research for some years, and the microbiome field started. So then I moved actually to the US, to Los Angeles. And we basically went back to that finding, and we started looking more deeply at what is there and using those deep sequencing technologies. So we looked at a region that is actually specific to fungi. And from there, it all started because that first few sequencing of intestinal specimens actually showed that there is a community of number of different fungi, which raised, as you can imagine, millions of questions. So it became very exciting. And that's how I went in that direction. And now actually all my lab is working on that. So
what creatures have you found a mycobiome in? People are you studying, or what kind of creatures?
So we have looked actually in our main goal is to look what is in humans, of course, because of the diseases that we are studying in my lab. But we have looked at different creatures. So we looked at many different mammals. We looked at dogs, rabbits, a lot of herbivore animals, pigs, goats, many animals actually. So the herbivores, they have a lot actually that are coming, a lot of fungi that are coming with the diet. And of course, we look in mice because those are the models that we use in the lab, and we can target the microbiome there.
So you've seen that pretty much all creatures have a fungal constituent, a microbiome?
That's right. It's very interesting. So some of them, of course, are associated with plants because a lot of fungi are actually on the plant surface, and they're in the roots of the plants. So that's a mycorrhiza, that's what's called. So it's a community of fungi that plants use to utilize nutrients, but animals eat those plants. So we can find some of those fungi, but what is more interesting is that we also find fungi that are not associated with plants or the diet. And that's what we are focusing a lot on.
Interesting. So I know like people get athlete's foot, or, I've heard they've got a fungal infection of their nail, or, something like that. So maybe that can be recast in the light of, again, we have fungi all the time. Are they throughout our entire body, you think? Or are they in just a few select areas like bacteria? I mean, bacteria are supposedly everywhere in our body, but are fungi as well?
That's right. So, for example, on the feet, that community is very well studied because of the infections that happen. And some other fungi are very well studied at different parts of the body because they call to very obvious infections. What hasn't been studied, though, are the fungi in the gut, because they don't lead to an infection that can be just easily detected. The gut is not accessible, and fungi were not looked in the gut. So that's the area where we mainly focused, and that's where we found this very wide community. But you're right, fungi are covering all our body surfaces, and as a mucosal immunologist, I'm interested in all of them because, as we will probably talk about later in our conversation, one side of the body can prime another side of the body, and where the organisms are and where the priming is happening is something very important to understand.
This sounds worse to me, I guess, on a gross level, because me being full of bacteria is one thing, but when I think of fungi, I think of like spores and hyphae and mats of growth and mushrooms, and I mean, I know it probably doesn't go that far, but I mean, well. Bacteria, maybe it does. I don't know. Bacteria make biofilms. I mean, what do fungi do inside of us?
Right. So a lot of it we don't understand, but it's definitely not gross. So we have a lot of images in the gut, and it's actually more structured than a gross picture. But imagine you are eating bread, you're eating cheese, you probably enjoy wine. So there are yeast components in all those foods. The beer, the yeast are fermenting those foods, basically. So it's not that horrible as one would imagine. But so what is in the gut is very interesting, and how those organisms actually survive in such a harsh environment. If you think of food products where fermentation is occurring, those are not such a complex system. But in the gut, where you have very hostile environment, you have a lot of bacteria that will compete. You have anaerobic situation where not a lot of where oxygen is actually not getting in. So how fungal organism will survive there? So those are very interesting questions. That's very fascinating about the microbiome.
Are there aerobic and anaerobic fungi? Or is it not classified like that?
Yeah, they are. So in humans, we haven't seen actually typical anaerobic fungi, but some of them are known to be in the ruminant animal's stomach. So basically cows, they have few stomachs, and one of those stomachs is actually fermenting a lot of plants and cellulose. So that's where you can find anaerobic fungi. In our gut, fungi are aerobic and facultative aerobes. So it's more complicated, yes.
Yeah, I know this guy. He's a DUI lawyer, and he sent me an article about auto brewer's syndrome. I guess like some people, they took antibiotics and it allowed yeast to grow in their guts, and the yeast would ferment, sugars and carbohydrates and stuff and make them drunk.
That's correct. That's actually real. I use it sometimes as the awakening call in my lectures to the students. But there are people that actually have that, and there are some reports, yes. So basically, fungi are embedded, probably in their stomach mucosa of what some of those reports are actually saying, but we never had a patient like that, so I cannot really tell. But yes, fermentation occurs when they consume sugars or even bread, so they have to be very careful. But it's very small population, but funny one.
Yeah, interesting. So what kind of structures do fungi make inside of creatures?
Well, so the way they live, they're single-cell organisms, but of course they can embed into biofilms. So they're very, very good examples for biofilms, for example, on infected skin, where also fungi can form them, and they form those matrices where the bacteria are actually embedded. There is a similar situation in some patients with disease called cystic fibrosis in the lung. But in the gut, we have done a lot of microscopy, and many others have done it. So we don't see actually those biofilms, or they're just not easily detectable in the gut. What we see in the gut is those patches basically of fungal cells that are embedded in the microbiome. So they're part of the microbiome, right? So we see them immersed in that mixture of bacteria and other substrates that are in the gut. And we see them in very close association with the mucus. So the mucosa is basically, the mucus is very close to the epithelial cells. So that's how all that connection probably is happening. So we investigate those processes.
Do you think that fungi help establish a niche for bacteria inside of us?
Yeah, this is very...
They work closely together?
Yeah, this is very interesting, branch of this type of science. So certainly there are examples of fungal-bacterial interactions, and very well so in the nature and in the soil, where some fungi produce antibiotics and create their own space in the bacterial niche, or vice versa. Some bacteria can affect the fungi, or some organisms, for example, there are ants that are basically using the fungi to ferment their own food. So that's where basically bacterial and fungal species are found in this coexistence, which is beneficial. So how is that working in the gut is very interesting, and it's also a very complex question. So we are approaching that question from a few different angles to start gaining basically more information. If you think that you have different species and even strains of fungi that are very interesting, and they're embedded in the bacteria, who will interact with who, you cannot predict that in such a complex system. So we are starting to learn more about the genetic information those organisms carry with them, and probably products that they can produce to signal to bacteria or to basically get bacteria out. So this is basically the current approach, but it's a very exciting field. There is a lot of potential there, I think.
Well, what would be a good model to study all these interactions? What about plants? I guess in their roots they have these nodules that are full of bacteria, but they also have fungi that interact with them. Are they a good model to study this, or are people better, or mice?
So yeah, plants are a great model. So, and they're studied extensively because of agricultural science, and now because of the carbon capture, which can be captured by basically in those fungi in the plants of the roots. And there is a lot of research on that. This very interesting perspective, but from our point of view, this is not the model that we are pursuing, because we are more interested actually what is in the gut of the patients. We are not even looking in a classical infection or in a classical symbiosis situation. We are looking at a situation where different individuals come here with different type of fungal communities. But some of them have a disease and some of them do not. So we have to get down to strain and species level to understand what is going on there. And from there, we have to link it to the patient condition and look how that is influencing their disease. So plants are a good model, but not for what we are doing. So our angle is a bit different. But yes, a lot of groups are interested in plants associated fungi.
So what are some of the specific fungi that you found inside of are you studying people, or are you dealing with mice, and what specific creatures have you found, and what appears to be their function?
Yeah, so in humans, we have found a lot of different fungi in different individuals. But what sticks as something very consistent is that Candida species are very prevalent in the human population. and some of them expand in patients with inflammatory bowel disease. So this is basically the focus. And those organisms, so we use deep sequencing technology so we can look at the DNA, but DNA can be coming from anywhere. So if you have fungus that is coming from the food but not sticking out there, you would detect the DNA. But when we link that to more complex system where we actually work with the live fungi we are getting from those from those individuals and from specimen from those individuals, that's where the picture becomes more comprehensive. But short answer to your question, yes, Candida species are the most prevalent species in the gut of the human population across many different studies, including our own research.
What do you think they're doing? Are they producing beneficial metabolites that are food for bacteria, for our cells? Are they maybe they're helping to digest things that bacteria and our cells cannot?
So Candida species are less interesting in terms of a secondary metabolites. So if you think, for example, of penicillium, where the penicillin is coming from, or other filamentous fungi such as aspergilli, they produce a lot of secondary metabolites. They have that program, and those secondary metabolites can have different antibacterial functions and so on. But if you look at Candida species, they actually carry a program that makes them very good survivals in harsh environments. And I don't know if evolutionary because of that or because of something else, they have dropped a lot of, they basically lack a lot of genes for secondary metabolites. So, but of course they produce metabolites because they have to live. So it might be that they produce metabolites that are more similar to our own systems and interfere with that. Another way they interact is directly basically interacting with immune cells, and that's the way how they prime our immune system. And we basically have been looking for cells that are interacting with fungi, and that was something we discovered a couple of years ago, a population of cells that is directly sensing fungi and can uptake them. So it can be direct interaction and more indirect interaction. Mixture of that is probably where the truth is. And if you put the other level of complication, which you also infer to basically how bacteria and fungi live together, and there might be ecological competition and all that, there is evidence for that. So, you know, the picture is basically more complex, but those three major components are basically what are shaping that community to live there, to stay there, and to influence host health and disease.
Well, I would think that if this is just my speculation, but if fungi are only found in a few places on our body, for instance, that would say, okay, maybe they have limited function. But if they're found everywhere, like bacteria are, that would mean to me that they have many, many functions that we may not even know about. Like, when I go to the bathroom, are there any fungi in my pee versus, if I poop? Poop, probably yes. Pee, I don't know. What about if I sneeze or if in my mouth, do I have fungi there that are part of my microbiome? Is it everywhere?
Right. So so there are fungi in the mouth, there are fungi in the intestines, and there are certainly fungi in the poop, and some of that is coming also from the diet. In the urine, depends. Some patients actually can develop fungal infections, and we are studying some phenomena like that. So we detect fungal DNA. Is that actually active fungi that are proliferating and living there is the key to understand how is that influencing those humans that have those infections. But yes, so fungi are all around. And if you think about that, right, so we have patient populations, and I will tell you more about like a specific study that we have done, but there are specific population of patients and subclasses of those patients that where we have evidence that fungi might be actually part of that pathophysiological process in those individuals. But if you think for the global population, if so many of us and also many animals carry fungi and they are normally associated with our body surfaces. So there might be another side of the story where fungi actually have beneficial role for the host. And we have few studies, but they are for now only in mice. So if we basically treat mice with antifungal drug for a long time and induce basically antifungal drug resistance. So fungi that are resistant to specific drugs, they will expand. We can see that those animals develop more severe intestinal inflammation and also inflammation in the gut distal sites, such as the lung. So that's something very amazing from an immunological point of view, how one organism that is in one site can affect actually immunity at other sites. So there are many different ways we probably play with fungi, and so much is unknown. It's a very exciting field to be in.
Yeah. What are some of the specific experiments or research that you're doing?
So there is something very exciting. So for some time, of course, a lot of antifungal receptors have been known because of the field of mycology and infectious diseases and fungal immunology, which would explore basically a situation where fungi would infect the host. So humans that are defective in specific proteins and genes, they would be more susceptible to fungal infections. But in the gut, we have been looking at what cells actually would detect the fungi, what cells would be actually carrying those specific receptors and have the capacity to interact with fungi. So about 2 years ago, we found a population of cells that express a chemokine receptor called CX3CR1. But that receptor actually have nothing to do with fungi. So we went to explore deeper, and it turned out that those cells are expressing a number of different molecules that are important for recognition of fungi. And those cells can actively actually engulf, phagocytose, the fungi in the gut. So from there, we went into a patient population of patients with intestinal disease called Crohn's disease, which is quite prevalent actually in our society, and it's a lifelong disease, chronic, very difficult to treat. And there are a lot of flares even if it's treated, and a lot of patients don't respond to medication. So in those patients, we found a mutation for the gene for this chemokine receptor, CX3CR1. And the patient that had this mutation lost a function allele, basically. They lost their ability to produce antifungal antibodies. So it's a small population inside of the IBD population, of Crohn's disease population. But are those the patients that actually are deficient in their ability to generate response to fungi? So should we treat that type of patients with antifungal drugs as a co-therapy for their intestinal inflammation? So you see, it's very interesting. It's very complex. It's not a classical infection that we are talking about, but it's more like a crosstalk between fungal organisms and the host immune system. So that was one example, very exciting findings that we have had, and we are pursuing in that direction.
Yeah, I mean, in terms of fungi, the only time I've ever heard about them in the body is in terms of an infection. I've never heard about them being talked about as being commensal. And, just like in bacteria, if someone has antibiotics and then, let's say, C. difficile takes over, does the same thing happen with fungi? If someone has a yeast infection, is that because it's an infection, or is that because they had that yeast the whole time and for some reason it was just able to grow out of control?
So it depends on the context. So in patients that are, for example, that have been treated with antibiotics for a long time, depletion of specific populations of bacteria can lead to expansion of specific fungi, and that's a documented phenomenon in both mice and humans. In patients that are also immunosuppressed, so bone marrow transplant patients and so on, the gut can be, for example, the site from where fungi transfer systemically, and they lead to deadly infections. So we carry those organisms, they are commensals, but in certain situations, they can be deadly. In other situations, they can be doing something that we don't know. So in the context of inflammatory bowel disease, nobody actually gets this systemic fungal infection. Nobody dies from fungal infection there, but fungi expand in the gut, and we have a lot of evidence now that they're priming basically the immune system. And priming the immune system in a situation of inflammation is something certainly not good. So is that actually specific fungi that are transferring? Is that even specific strains that are transferring from one human to another, or are they just living there? So those are very important questions to answer in terms to actually develop future therapeutics in that area.
Is there a lot of interest in this area, or is all the interest being sucked up by bacteria right now?
A lot of interest is sucked up by bacteria, but actually there is growing interest in fungi now. And more groups that are doing traditional research in bacteria or immunology are also interested now in fungi. We and others developed a lot of tools actually to work in this area. So I think the most interesting discoveries will just happen in the next years. The more the merrier, basically. So there is interest. Less interest actually on the industry side, but because those things they usually lag, right? So that will come as well, I believe.
I don't know if you were to look at bacteria versus fungi. Do bacteria have a more privileged place, or do they seem to have more effect in the humans, for instance, versus fungi, or it's just you can't really say that. They just both have their own effects. I mean,
yeah, so
one more important than the other. Yeah.
Right. So if you look globally, right, fungi as a kingdom and bacteria as a kingdom, of course, the bacteria are more, for example, in the gut. But what we are learning is that actually we need those bacteria. They're not bad for us. So they're bad players, which are basically specific species and even strains. Not all the same species actually carry genes that will be detrimental. It's certain strains that, for example, can acquire antibiotic resistance gene, and then that's a problem during disease. So it's very similar, I think, for the fungi. So they are just we don't know it yet. So that's where we are actually diving right now to basically distinguish what species and what specific strains are actually detrimental. So you cannot tell it's basically all the fungi or all the bacteria. There is something that is less mythological, if you wish. There is actually a carrier of phenotype in many cases. So it's more complex because it's a community. So one change can lead to a number of changes downstream, but what is the primary cause is something that's very important. And I think here a lot will be basically specific organisms. It wouldn't be as mythological, as broad as, the mycobiome or the microbiome. It will be specific organisms and specific pathways and genetic capabilities that they carry with them.
Is it? I don't really know much about fungi at all. I mean, what is it? What are their cells like? Do they have their own DNA, and have they been sequenced? Is it hard to sequence them?
Yeah. So the cells are actually bigger. They're from 10 to 100 times bigger than a bacterial cell, and they carry very rigid cell walls. So it's hard to extract the fungal DNA from there. But, the field is getting in that direction. So now the microbiome studies, number of groups now are actually doing also the specific fungal extraction. So they don't want them to miss that information because that's actually the microbiome, right? You cannot miss such an important component. So the cells are bigger. The cells are very interesting because they have specific polymers, and there is a repertoire of human and mammalian receptors that can recognize actually those different components. And if you go evolutionary down and you go, if you go to plants, if you go to lower organisms, all of those actually have some type of systems and receptors that can recognize fungal components. So you can see how evolutionary actually fungi are very important for immunity or protection or sensing by organisms. So they're bigger cells, they're very interesting cells, they can produce a lot of secondary metabolites, and they have specific cell walls. So from immunological perspective, it's also very interesting because you're actually now tapping on pathways that are not that commonly activated by bacteria. And that's also our hope. That's what we are trying to track in the host to actually understand when antifungal immunity is actually on. And in mice, we can do it. The challenge is in humans, and that's where we are going.
Very interesting. So what's the best way for people to learn more? I mean, it's not, I don't know much. I've talked to a lot of people, but people listening, same thing. So what's your recommendation on how they can learn more?
So people should look actually in specific, they should basically look into the main literature, or nowadays I actually see more and more reports in journals, in articles for fungi that are basically not just pure scientific information, but more digestible for the general audience. And all the groups, they have their websites, and every one of us is trying now to communicate science to the broader audience. And I hope people can learn more by this approach. But again, all the space is flooded by information about the microbiome, and it's getting there that basically it sounds that the microbiome is involved in everything, and people start actually losing interest because if they say, okay, I have any disease, it's probably also microbiome related, but people should look actually for more specific information, and that's where the gem is actually. So that's what I think.
I guess if people knew the actual complexity of what's going on, I mean, they know. They just can't look at it all. It's like so overwhelming.
Yes.
Freak out and not do any science, So you have to
shield
your eyes.
Right. But it's not about the scientists. The scientists, each one is having a preferred area, and they are actually very, they're diving deeply into the problems in that area. It's more about, the general public. So how do you basically understand the microbiome? What do you know about it? It's not that, okay, the microbiome is involved in heart disease, in IBD, and all those diseases, but what are the components of the microbiome? What is the new science that is coming? That microbiome is involved in everything. That's an old news. So now we have to know where specifically and how. So that's where I think the field is heading towards.
Well, very good, Iliyan. Thank you for coming, and I appreciate you being here.
Thank you, Rich. That was a pleasure.
You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you.